Welcome, M1s. I am absolutely thrilled to guide you through today's explainer. We're going to be taking a really fascinating journey from the incredibly elegant, tightly controlled world of normal cellular homeostasis straight into what is essentially a complete molecular system failure. And, well, that's what we clinically define as cancer. By the end of this, you are going to understand exactly how cells manage to escape their normal controls, completely ignore biological boundaries, and invade surrounding tissues. So let's dive right in. Okay, here's our game plan for today. We're starting with the normal cell cycle and its built-in fail-safes. Then, we'll shift gears to the security breach itself, comparing oncogenes with tumor suppressors and seeing exactly how cancer gains its survival advantage. And finally, we'll tie the whole shebang together by looking at sporadic versus inherited cancers, capping it off with a classic clinical case of retinoblastoma. Section 1. The normal cell cycle. Now, before we can really grasp how the system breaks down, we absolutely have to appreciate the strict regulatory controls that keep our cells behaving day in and day out. Think of the cell cycle checkpoints as these highly secure, non-negotiable toll booths. Considering your body replaces billions of cells every single day, these pauses, they're literally a matter of life and death. Take the DNA damage checkpoint. It actively slams the brakes on the cycle until any faulty DNA is fixed. Or, over at mitosis, you've got the spindle assembly checkpoint asking, hey, is the spindle fully built? Are the chromosomes aligned properly? These vital pauses are exactly what prevent faulty DNA from snowballing into unregulated growth. Beyond those structural toll booths, there are three primary mechanisms pulling the strings here. First, you've got your internal checkpoints. Second, the physical length of the telomeres. We're going to unpack that biological ticking clock in just a minute. And third, there's this highly orchestrated chemical symphony going on. We're talking cyclins and kinases that kick off mitosis, working alongside various hormones and localized growth factors. Section 2. Apoptosis and system failsafes. So what happens when the DNA damage is just way too severe to repair? Well, the cell basically has to fall on its sword for the good of the organism. And that brings us to a massively important definition for your boards: caspases. These are, without a doubt, the executioners of the cell. When a cell is officially doomed, caspase enzymes step up to violently carry out the destruction. We're talking collapsing the nucleus, condensing the chromatin, totally shutting down DNA repair, and completely dismantling the cytoskeleton. You might be wondering, how does this physical destruction actually play out? It's a pretty wild four-step process. First off, a death signal docks at a specific cell surface receptor. Second, those executioner caspases we just talked about get activated inside. Third, the cell physically starts undulating. It literally breaks apart into these little fragments we call blebs. And finally, phagocytes sweep in like a cleanup crew to engulf and degrade whatever's left. You can actually visualize this exact biochemical pathway unfolding physically right here. The death receptor grabs the signal molecule, then the caspases light up internally, and boom, the doomed cell breaks apart into those tiny, easily digestible blebs right before being engulfed. It's incredibly efficient. The threat is just neatly and cleanly removed. Section 3. Oncogenes versus tumor suppressors. Okay, so we've seen the normal controls and the failsafes. Now, let's look at the actual security breach. Because fundamentally, cancer is a genetic disorder. It's caused by mutations in the very genes that govern those exact pathways. To really get cancer genetics, you need to contrast two distinct types of genes. Think of them as the gas pedal and the brakes. First up, oncogenes. These are the gas pedal. When they mutate, it's like a brick gets dropped on the pedal. They get stuck in the down position, promoting completely uncontrolled division. On the flip side, we have our brakes, the tumor suppressors. Normally, they trigger apoptosis, but when they are mutated, the brake lines are cut and the cell totally fails to halt the cycle. Let's look at a concrete example. Check out the normal RAS proto-oncogene. It's doing its job, appropriately responding to a growth factor and promoting division when asked. But then, look at the mutated RAS gene product. It is physically locked into its GTP binding shape. It's completely jammed in the on position. And what does that do? It causes this incredibly dangerous, relentless proliferation, even without any external growth signal telling it to do so. Now let's zoom in on those brakes, the tumor suppressors. As future physicians, you absolutely must know these high-yield genes. p53 is your classic transcription factor that steps in to promote apoptosis for damaged DNA. RB, on the other hand, halts the cell cycle right in the G1 phase. And BRCA1, that one repairs DNA sequence mutations. If you inactivate these, the cell literally loses its primary defenses. The whole system is left wide open. Section 4. Gaining the survival advantage.
So the genetic operating system has been preached. How do these malignant cells actually manage to outcompete and survive over our healthy cells? Well, it starts with the number 50. 50 is the absolute natural mitotic limit for human cells. You see, telomeres, those little protective caps at the ends of our chromosomes, they physically shorten every single time a cell divides. After exactly 50 divisions, that shortened length triggers a hard stop. It permanently shuts down mitosis.
But here's where it gets crazy. Cancer cells actually hack this biological clock. They use an enzyme called telomerase. Telomerase actively rebuilds and elongates those telomeres. So what does that mean? It means a cancer cell completely bypasses that 50 division limit. It achieves functional immortality, which, by the way, is exactly how those infamous cancer cell lines, like HeLa cells, have managed to just keep dividing in research labs for decades on end.
And immortality isn't their only weapon, not by a long shot. These cells take on some seriously rogue attributes. For one, they lose contact inhibition. So instead of stopping when things get crowded, they just pile up into dense masses. They also become highly invasive. And perhaps most remarkably, they undergo angiogenesis. When these growing tumors start starving for nutrients, they literally hijack the body's own infrastructure. They force the growth of brand new blood vessels just to feed themselves. It's terrifyingly clever. Let's look at the macroscopic reality of these microscopic advantages. Tracking this abnormal lung tissue, you start with healthy, happy, ciliated cells at the top. But as mutations start to accumulate, let's say years after someone starts smoking, those cilia are destroyed. The cells start to flatten out, they pile up into atypical masses, and eventually they completely breach the basement membrane. At this point, they're undifferentiated, they're rapidly dividing, and they are highly, highly invasive. Section 5. Sporadic versus inherited cancers.
Now let's zoom out from the tissue level and look at the actual patient. We need to understand how these molecular mutations translate into clinical susceptibility for the folks you're going to be treating. Notice the really stark visual distinction here between germline and sporadic origins. With a germline, or inherited susceptibility, the patient has that first mutation embedded in literally every single cell of their body. But with sporadic cancer, it begins with a completely spontaneous, localized somatic mutation in just one specific little area of tissue.
So what this means in practice is that for inherited cancers, every cell has this baseline risk. But cancer usually only kicks off when a secondary mutation happens. So it runs in the family, but it might present as entirely different types of cancer, depending on exactly where that second mutation decides to strike. Sporadic cancers, on the other hand, arise purely in somatic, non-germline tissue. These are spontaneous errors, and the risk is often driven up by exposure to environmental carcinogens, things like UV radiation or chemical toxins. Section 6. Retinoblastoma, a clinical case. All right, let's synthesize all this dense molecular and genetic data into a practical, board-relevant clinical vignette. So, here's the clinical scenario. Now, cancer rarely follows strict Mendelian inheritance, but a few specific cases actually do. Imagine you've got a pediatric patient presenting to your clinic who inherited a mutation in the RB gene. Remember from a few minutes ago? The normal RB gene is a vital tumor suppressor. Its job is to halt mitosis in the G1 phase. This patient is a perfect textbook illustration of the classic two-hit hypothesis. That inherited RB mutation, that's the first hit. It means every single cell in this child is already susceptible. However, the actual retinoblastoma will only develop if and when a localized, sporadic secondary mutation, the second hit, strikes the one remaining healthy copy of the RB gene in the retinal tissue. Once that happens, both sets of brakes are gone and cellular control is completely lost. Which brings us to our final and honestly most important question for you as future physicians. How might understanding this incredibly precise molecular loss of control, the locked RAS proteins, the hijacked telomerase, how might that lead to targeted clinical therapies? Think about it. If you know exactly how and where the system fails, you literally hold the keys to designing the pharmacology that can stop it in its tracks. Definitely keep that in mind as you continue studying. Thank you so much for joining me for this explainer, and I will see you next time.